All right. Good morning, everyone. I'm Rashmi Kanagal Shamanna. I'm a professor, board certified in molecular genetic pathology and hematopathology at MD Anderson Cancer Center. I also co-direct both the bone marrow laboratory as well as the innovative diagnostics section of the clinical molecular diagnostic laboratory. Thank you very much for inviting me, and it's a pleasure to be here. Uh, in this session, I will be presenting expert recommendations on behalf of the International Consortium for Optical Genome Mapping for integrating OGM as a valuable clinical research tool into cytogenomic and molecular workup of hematologic malignancies. So these recommendations reflect the work by multiple international experts across the world who have extensive OGM experience across different hematologic categories as well as different laboratory settings. So as we all know, accurate cytogenetic profiling with comprehensive characterization is really central to heme malignancy research. It can help identify underlying pathogenic mechanisms as well as potentially provide guidance for clinical trial enrollment and potentially change management as well. So we know the traditional methods that includes G-banding karyotyping, FISH, chromosomal microarray, they all have inherent design and resolution limitations. So many of the cryptic or complex SVs that might be important may remain undetected, and that might contribute to incomplete characterization and also lead to inter-laboratory variability in various research settings. So that's where OGM is key, right? So as we all know, it's a high-resolution genome-wide platform that is able to identify major classes of structure variants, copy number alterations, balance rearrangements, and complex alterations, all within a single framework. So the resolution of OGM exceeds that of conventional karyotyping, and it very nicely complements the results from sequencing-based assays that provides a more comprehensive genomic profile for any investigation. So heme malignancies, the studies on heme malignancies, they benefit because there's abundant bone marrow and peripheral blood samples available. And we all know that many peer-reviewed scientific reports have already demonstrated superior success rates where this valuable research tool is able to identify many cryptic SVs compared to the conventional counterparts. So the most important thing is OGM offers this unbiased genome-wide coverage without knowledge of any prior probe design. So that's been already implemented in many, many research labs, particularly because it can streamline many of the assays, consolidate into one. Which is crucial for clinical research. However, despite this growing evidence, the adoption into clinical research workflows, it's been variable. The key challenge here is that it lacks some sort of recommendations regarding the standardization of the assay for evaluation, regarding what indications and in what heme malignancy subtypes this might be proved to be useful. Just the sheer amount of data complexity, the complexity in interpretation, and also the resource considerations. So to address this, the ICOGM collaborated with an international team of experts who are cytogenomic, molecular, and clinical research in respective fields. And this spanned across different institutions across the United States, Canada, Europe, and beyond. So the consortium addressed these questions with practical guidance on when and how to implement this as a novel tool for research and how to interpret the results and etc. So the recommendations were two-pronged. So the first set of recommendations was focused on general consensus on specimen requirements. Obviously, as we are all aware, bone marrow is a preferred sample compared to peripheral blood, but there are some exceptions where so if there is enough circulating blasts in the peripheral blood and bone marrow is not available, then peripheral blood will be a very optimal specimen as well. In addition, in settings that might have fibrosis or bone marrow hypocellularity, in fact, peripheral blood, because it's more representative and has a higher number of cells, may turn out to be a better sample. So the for preservative, EDTA is definitely preferred over heparin. It is important that these samples, the OGM assay is done within 72 hours at least uh, of sample procurement, and ideally within 24 to 48 hours, but no more than 72 hours. And if anyone wants to kind of use it later, consider OGM at a different time point to explore, then these cells could be preserved at minus 80 degrees. And in certain scenarios, of course, considering the limit of detection of OGM, it may be helpful to enrich for specific cell types. For example, CD138 enrichment in plasma cell neoplasms, or if you want to look at abnormalities in myeloid malignancies, um, we might enrich for the myeloid neoplasms after sorting. So the second set of recommendations were actually disease specific, where we kind of looked into different entities, heme malignancies, primarily involving bone marrow or peripheral blood. So in general, the OGM it can be a very first line research assay in conjunction with the other conventional tools uh, in most of these cases. Or it could also be it could be used as a second line approach. That, of course, depends on the laboratory resources. Important to keep in mind that if it's used as a second line strategy, OGM is very informative, specifically when the karyotyping cultures are inadequate or they fail, or when a conventional G-banding karyotype is normal, or when the cytogenetic findings are a bit ambiguous, or in rare malignancy, right, where OGM can serve as a very good tool for discovering new abnormalities. In any case, it's important to preserve some of the leftover material for potential downstream analysis in order to confirm any of the novel findings that might be detected using OGM. So here's the next set of disease-specific recommendations. So the recommendations spanned both adult and pediatric hematologic malignancies that involved marrow or the blood. That's AML, MDS, MDS-MPN, myeloproliferative neoplasms, eosinophilia-associated neoplasms, as well as lymphoid malignancies like B and T ALL, CLLs, plasma cell neoplasms. So if resources limit universal use of OGM, it is important that the samples that are prioritized would be those where the karyotype has failed, or there's no cultures, or when there is insufficient metaphases less than 20, or in cases that lacked recurrent genetic abnormalities that is seen in these neoplasms by conventional tools. So in AML, we can go subtype by subtype. So in AML research, so OGM identifies a recurrent as well as rare rearrangements like KMT2A, MECOM, some of the rare RARA variants, and it's able to clarify these complex chromosomal architecture. Especially, it provides additional information when TP53 alterations are present, right? It can inform us the allelic context by identifying cryptic deletions or even copy neutral LOH involving TP53. So Inappropriate instances orthogonal methods can be used for confirmation as well. So OGM can also be helpful in AML when conventional data is suggesting low complexity, but yet we are suspecting additional structure variants like NUP98, for example, rearrangements with NUP98 when you see WT1 mutations by sequencing. So in MDS research, OGM has definitely enhances detection of novel genomic alterations that have been shown to be very important. For example, KMT2A partial tandem duplications, TP53 deletions, and copy neutral LOH, where the status of the TP53 makes a difference. It's especially useful in hypoplastic samples, right? Hypocellular samples, because you cannot grow them that easily. In MDS-MPN, OGM also helps concurrent assessment of many of the rearrangements that are referenced in various classification systems like BCR-ABL, PDGFRA, PDGFRB, FGFR1, JAK2 rearrangement, etc. So in specifically when it comes to marrow hypocellularity in conditions like aplastic anemia, in bone marrow failure research, OGM can actually help delineate those clonal structural alterations at very high resolutions in order to separate them from other non-clonal processes. Similarly, structural variants that might be relevant in germline predisposition, they may also be revealed. So for myeloid lymphoid neoplasms with tyrosine kinase fusion, it's really helpful because it allows for unbiased detection of any kind of TKI fusion event, and it's all done using a single platform, right? It reduces reliance on multiple targeted FISH panels, panels after panels, especially the cascade algorithms, and it kind of consolidates the research workflow and improves resources as well. Finally, in myeloproliferative neoplasms. So in myelofibrosis and other conditions associated with fibrosis like post-PV or ET myelofibrosis, particularly when the myelofibrosis grade is 2 or over, OGM is very helpful because the cultures are unsuccessful and the peripheral blood is actually preferred because the bone marrow may be a dry tap. So OGM in that case can actually detect genome-wide structure variant assessment, which and PB is more representative. However, in myeloproliferative neoplasms that do not have significant amount of fibrosis, OGM has limited utility. Now let's move on to lymphoid neoplasms. So in BALL and TALL research, OGM is able to provide this comprehensive picture of all the structural variants, and that's where it's made the biggest impact because the results are able to complement the classification aligned research workflows. So if we look at BALL, for example, OGM is able to identify recurrent translocations like BCR-ABL, ETV6, RUNX1, or KMT2A rearrangements, IGH translocations, as well as the pH-like associated events. Like there's a whole list of pH-like that includes CRLF2 rearrangements, ABL class fusions, alterations involving JAK-STAT pathway, the IM21, the ploidy changes, etc. In some cases, even small deletions, such as those involving IKZF1 and CDKN2A and 2B, they are also accessible. So in TALL research, so again, the same impact, right? OGM is able to identify all of these class-defining genetic features, like those involving BCL11B, some of these chimeric protein fusions, the KMT2A rearrangements with the appropriate partners, etc. And if you look at the other lymphoid neoplasms that involve the bone marrow, for example, CLL, OGM is an alternative assay to assess TP53 deletions, and it adds additional info on genomic complexity as well if you want to explore further. The plasma cell neoplasms, the FISH after CD138 enrichment is usually the standard way to look for some of these abnormalities. But OGM is a very viable alternative clinical research tool. And especially when there's abundant of plasma cells, we are able to interrogate many of these high-risk abnormalities. In pediatric research, in pediatric malignancies, whether it's AML, BALL, or TALL, there's a ton of evidence published in the literature that supports OGM as a superior assay for clinical research, given the high frequency of structure variations in pediatric leukemias and the value of this comprehensive genomic characterization up front. So it essentially, to summarize the findings, the consensus recommendations from the ICOGM support standardized integration of OGM as a valuable tool into clinical research workflows. And OGM provides equivalent or even superior SV detection ability compared to the conventional cytogenetic approaches. And at the same time, it has this ability to consolidate multiple assays, and it can very nicely complement sequencing findings as well. And it's very important to standardize this because it enables standard comparison across multiple sites that would lead to a higher success rate in clinical research. Further, prospective exploratory research in the setting of clinical trials can also help further refine future classification frameworks. Now, with that, I would like to once again acknowledge all the experts for their efforts to the ICOGM in generating these summary documents, and that's available as a publication in the American Journal of Hematology. So thank you for your attention, and with that, I am open to questions.